Okay, thank you very much to Professor Alfredo Martinez for her kind words and also for his invitation here to be here with you today. And good morning to everyone. I think this is a nice topic that we can share this morning, and we will try to have a good time and learn some things about this new field of nutrigenetics. Okay, this topic today is Nutrigenetics and Mediterranean diet, and I will have my talk. We had 4 parts, small one, a little bit of background, then Mediterranean diet and obesity, some examples from our lab in this field, and then some conclusion. Okay, I know that you are all familiar with this idea that now there is a complex relationship between the health of the individuals, its genome, and also lifelong dietary exposure. And nowadays we are learning more about this, and we are conscious that nutrition is among the most profound of all the environmental factors which interact with our genome. Times ago, we usually say that genes are kept the same over generation, but that sometimes diet is changing a lot these days in this new generation. But we are learning more, and now we are conscious that there are also many genetic variations that are also modifying these ideas. And there is no doubt that nutrition is a gene-environment interaction science, and there are 2 things that we have to take into consideration. We have to take into consideration genetic susceptibility, but also the exposure to the environment. And until recently, we usually have these dietary recommendations, and we assume that all individuals were identical culturally, ethnically, genetically, but this is not true, and we have to learn more about this. So we are starting a new era in nutrition, because now we can have the possibilities to direct our interventions to particular genetic profile. We also know that there are components of the diet that they are able to alter gene expression, and we also can go deeper in the understanding of the consequences of these genetic variants. So technology helps us also with this, and we are now able to study the nutrition, all the food components and nutrients that at a molecular level we can study them, and we can see how they interact over the risk of diseases. We may know the impact of these nutrients on genome, and also the result on the risk of diseases, and also the impact on lifestyle modifications. So now we are in this new field of molecular nutrition, and disease are taken in a new perspective, because we may realize that there are faulty genes that result in this dysfunction, and also this important interaction with the environment. There are some genes that are just explain the whole disease, but also there are also many factors, the environmental factors, that they are taking an important part in this developing disease risk. And particularly, we are here to study nutrigenetics, which is the study of the effect of genetic variant on the interaction between diet and health. In this interaction between diet and health, we have to take into consideration the genetic profile. We have learned a lot about human genome. Now we also have building new maps about haplotypes map or SNP variation maps. We are learning a lot in this topic. We may remember that this genetic variation can be mutations, deletions, translocation, inversion, or just replacement when a nucleotide is put in the site of the other one. This human variation, we now can be aware that this human variation, we have, as in the majority of cases, the same nucleotide basis, but this phenotypic variation at the gene level is about 1%, 0.1%, but there are millions of locations that can be changed, and these differences in human has this genetic basis because there are gene variations, many of them, millions or thousands of them. And this variation can affect our response to nutrition. So that's why it's important to study. And maybe we have a group of individuals with a particular mutation that has a different response to diet. We will study later that there are individuals with particular gene variations that have a response, different response, for instance, to Mediterranean diet. So, just starting with the second part, Mediterranean diet, we may consider that we are always in the prevention, disease prevention science, because we are in nutrition. And with this Mediterranean diet, as you know, many of you will know that this is the traditional eating patterns of the Mediterranean countries, the Mediterranean bordering areas, that they are known because there is areas in which olive is growing, these olive trees are growing, and initially it was the eating pattern of Crete, Greece and southern Italy. In the late 50s and early 60s, the origin of the Mediterranean diet. And it's based in these three important issues: wheat culture, olive trees, and grapevine. It's a mixture because there are at least 16 countries that are around the Mediterranean area. And it was a culture, they are different, just cultural, historical, or anthropological. And this is a great mixture. And this Mediterranean diet is not just a diet, as you know, it's lifestyle. And it's characterized by this plenty of food for cooking, never hurry up, this is a kind of philosophy, tasting food, space for friendship, pleasant meals, family meals, accompanying dishes with friends and time, and also regular physical activity. It's just not a diet, it's more a lifestyle. It's very important, the social component, and also the daily physical activity recommendation, daily physical activity practices. And this Mediterranean diet, you know, this is the father, is considered the father of the Mediterranean diet. Many of you will know him, is Angel Case, was a physician. In fact, he was a physiologist, as myself, a physiologist. It was time ago, 1957, when he observed that in 7 countries there was differences in diet, but also there were differences in cardiovascular incidence. And for the first time, they observed that there was a correlation. There were lower rates of cardiovascular disease when the people have a high dietary intake from fat. About 40% of energy becoming from fat. It was just mostly, as you know, not animal fat, but vegetal, mostly olive oil and vegetable. So this was the beginning of this idea that diet may prevent cardiovascular disease. This is also very well known, the Lion Diet Heart Study, 2001. It was the first clinical trial to study Mediterranean diet and to show a benefit for coronary events and mortality. It was performed of about 600 patients and for more than 2 years of following, and this nice figure that there was a high protection for coronary events and also against mortality. Okay, it was in 1993 when the World Health Organization put an ace on Mediterranean diet. There were an international conference and they tried to set up the basis for this Mediterranean diet, best traditional diet. And as you know, the most important thing here is that there is a low consume of animal products, low consume of animal products, and high intake of fat, 25 to 35 of calories, total calories from fat, and of course olive oil, better to say extra virgin olive oil, as the principal fat. So also previously in 2000 there was an international conference in London and they tried to set all the general principles for this Mediterranean diet. And here is a summary of these main traits of this Mediterranean diet. As you all know, abundant food from vegetal origin, using fruit as daily dessert, moderate consumption of milk-derived food, also a limitation in the consumption of red meat, and moderate consumption of wine during meals, as you all know. Okay, as also just to mention that Mediterranean diet is now in the least possible candidate to include in the UNESCO Intangible Cultural Heritage of Humanity. So because it's associated with a good health status. It's a very healthy diet, as you know. And what is the evidence for protection against cardiovascular disease of this diet? Pues, as you may know, many of these food items are now confirmed to be very, very well. Is able for all of these components of the Mediterranean diet, oleic acid, fiber, folate, flavonoids, all of them are able to lower this risk for cardiovascular disease. This is now confirmed that all of these food items are able to reduce cardiovascular risk. We have some of them, we are still not confirmed. There is no association with the elements that we have today with the works research. It's not enough. for this, but also we know some of food items that are not in the Mediterranean diet, that they are always increasing risk as saturated fatty acid, trans fat, sodium in excess, overweight, alcohol in excess, cholesterol. You may know all of this. And the important thing of the Mediterranean diet is this, is a eating pattern. It's not just to eat 3 food items, but a pattern. And this is the pyramid that you may know. And what may call your attention is that red meat are on the top of this pyramid, on the top, following by sweets, eggs, potatoes, and then dairy products are upstairs of olive oil, and of course in the base of the pyramid, cereal, fruits, and vegetables, as you may know. Okay. The important thing is that all of them has to be consumed together. So it's a it's an alimentary pattern. So just to stress the importance of extra virgin olive oil that allows you to eat more vegetables in a palatable manner. This is very important, the presence of the olive oil, because most of the benefits of this diet is because of the components of this oil. And here we will pass through Mediterranean diet to weight gain. We are here in a picture that we associate Mediterranean diet and weight gain. Okay, there is a hypothesis that many of the benefits of this Mediterranean diet on cardiovascular risk is due to this lowering effect on body weight. Many of this effect may be through lower body gain. So this is very interesting, because initially a diet that has a high content of energy from fat, you may think that it's going to get weight, the persons, the people will get weight, and it's not this idea. Here there are some studies that has a control group against Mediterranean diet, and some authors follow the individuals, the subjects, from 1 year to 2.5 years, and they study body weight differences between the control group and the Mediterranean diet group. And in most of these studies, there is a significant lower body weight, sometimes also significant lower BMI, a decrease in BMI, or decrease in body weight, 2 years, mostly in these studies of 2 years. The number of subjects is not so high, it's just less than 300, but it's interesting. These observations are still interesting. So, seeing these ideas, we just were designing a study with more subjects to see if this is true. Because as you remember, obesity is an excess of body weight, especially fat, and is accompanied for many other morbidities. There are also some association with this depression, anxiety, all kinds of alterations, medical alterations, mechanical, mental, and also more importantly, metabolic, diabetes, dyslipidemia, fat liver, hypertension, as you may know. So we try to study in genes related to body weight, how is the response to Mediterranean diet. Here there is a picture of how is the body weight regulation. And the systems, as you may know, there are three main systems that control body weight. Energy intake, all the proteins and the factors that regulate energy intake in the hypothalamus mainly. There is a large list. This is the more important component for body weight regulation, all these proteins and receptors that are in the hypothalamus. We also may be interested in the proteins that regulate energy expenditure. And also we have a large number also of genes that are involved in adipogenesis, in the growing of the adipose tissue, such as adiponectin or fatty acid binding protein, interleukin-6, PPR gamma. And some of these genes present gene variants that could change or modify obesity risk. So we are now starting the third part of the talk, and there we will see some examples in this Nutrigenetics file. So our study was a sub-project for the PREDIMED study. The PREDIMED, as the name says, is a prevention study for coronary disease in Spain, involving about 12,000 subjects, and with Mediterranean diet. And a subsample of this project, we have a study, that it was a nutrigenetic study, with the aim of studying genes implicated in corporal adiposity, the response of individuals to Mediterranean diet when they have several mutations in genes related to adiposity. So we studied these subjects for 2 or 3 years, follow-up study, and we study this response to Mediterranean diet, and also depending on genetic variations. As I told you before, the PREDIMED studies concern just subjects with high cardiovascular risk. So this PREDIMED study is a controlled, multi-center, randomized, prospective study in Spain in more than 8 cities with about 12,000 people followed for 4 years. And we studied some of these gene variants. This is the list of gene variants that we are studying. PPR gamma, proline, replacement by alanine in the position 12. For interleukin-6, there is a change in the position, in the promoter region, 174. There was a replacement of a guanine for a cytosine. In the adiponectin gene, we studied 3 variants, plus 45, replacement of a thymidine by guanidine, and another one in the region of the position of 276, and in the promoter region of this gene, important gene, long away to the initial codon for transcription, a replacement of an adenine by a cytosine. And we also studied this gene, the FTO gene. The FTO gene is a gene that has to do with the food intake and obesity. And this is the SNPs that is most extensively studied and implicated in adiposity. So I am going to present now our work with this Mediterranean diet. We have around 1,000 people, and we try to, we divide them in 3 experimental groups. One, it was control group that follow a low-fat diet according to the American recommendations. Then 2 groups, they were encouraged to follow a Mediterranean diet, this eating pattern that we have spoke before. One with an enrichment in extra virgin olive oil. The patients or the subjects, they were freely given a 1 liter per week. And then we have another group with Mediterranean diet, but a high content of nuts. 30 grams per day, walnuts, hazelnuts, and almonds. And these subjects were carefully followed by dietitians. Some of them maybe are here, or just to, and they were just helped them to cook according to this Mediterranean pattern. And if you want to look for information in this interesting trial, you may go to the web page of the PREDIMED study. Here I just will tell you about our small project within the frame of the PREDIMED study. And for our study design, we had this basal measure that at basal we measure many parameters in the blood, in the urine. We perform also electrocardiogram, and we have also the subject has to fulfill food frequency questionnaire and also physical activity questionnaire. We gather all this information. And for the inclusion criteria of the PREDIMED study, I just have to remember you that they were patients with high cardiovascular risk. To define high cardiovascular risk, the patient should have type 2 diabetes or 3 or more of the following risk factors. 3 or more of the following risk factors: habitual smoker, high blood pressure, LDL cholesterol higher than 160, or HDL cholesterol lower than 40 milligrams per deciliter. They has to be overweight or obese, and they also may have a family history of early ischemic cardiomyopathy. They were before they have a coronary event. So the patient has type 2 diabetes or 3 or more of these risk factors, just for us to understand that they were patients in high cardiovascular risk. And our study were focused in these 1,055 subjects. 92% of them were overweight or obese, and 37 were diabetes, with people with diagnosis of diabetes. And we perform also some anthropometric measurements, and we follow this patient for 2 years and 3 years, and we again perform, we again request the patient to fulfill food frequency questionnaire and physical activities, and we perform some blood and urine analysis. And this is important to understand our later results. This is the statistical analysis. With this study, in this study we perform just general linear models for regression and multivariate linear regression models. We had these two analytical studies, And it's important to understand that our dependent variable was changed in corporal adiposity. We study the magnitude of change over 3 years, over 2 years, body weight, or waist circumference, or BMI. And the dependent variable was the 2 years body weight, for instance, minus the basal body weight. It was change in corporal adiposity. And our independent variables were just of 2 types: adjustment variables, access, age, physical activity, and also we have effect variables. Effect variables that help us to understand the outcome of the result. So we studied this gene that you may know or not. This is the name is written there. This is a receptor that is expressed in adipose tissue that participates in adipose tissue differentiation, transcription factor that is very important for genes that are related to fat and glucose metabolism. And is involved also in insulin sensitivity. So here the variant that we were studying is a replacement of proline by alanine in the position 12. So our first gene variant study, our objective was to analyze the effect of both Mediterranean diet, followed by 2 years, and the polymorphism of this gene, that is a replacement of a proline for an alanine at position 12. For this study, we follow the 100 subjects, but some of them were not at 2 years available, and we have a total of 774 subjects that they were split in 3 groups, as we commented before: control group, Mediterranean diet plus virgin olive oil, there are more than 300 people there, and also 300 in the groups of nuts with Mediterranean diet. We study mostly waist circumferences, weight and BMI, basal and after 2 years. And we perform this gene mutation by these methods, the simple method, that a PCR and a digestion with a restriction enzyme. And these are the basal characteristics according to the intervention group. And these are the control group, the Mediterranean plus olive oil, Mediterranean diet plus nuts. And these are the people with the mutation. They are alanine carriers of this polymorphism, and they were just less of them, of course, than the white people without having this alanine allele. And for all these factors that we study, blood pressure, diabetes, smoking habit, we have no differences, no statistically significant differences between groups. All of them were similar in their basal characteristic. But when we study the chain, now we study the chain, the differences in waist circumference, as you remember, the differences between 2 years waist circumference and basal. And what we see is that ALA carriers, subjects with this mutation, they have higher amount in this magnitude, higher chain, a positive magnitude for change. that it was statistically significant. So they have near 1 centimeter higher waist circumference. This is waist circumference change, and it was just near 1 centimeter. When you have the mutation, your waist circumference is 1 centimeter higher. So having the mutation is modifying your waist circumference change. And as you may see, there was no effect, no differences, significant differences in the Mediterranean, in the nutritional intervention groups, was the same. And also, in this model, we obtained that basal BMI was subjects with high basal BMI has lower waist circumference change. This is also an important thing. In this regression model, people with high basal BMI has lower waist circumference change. A small decrease in the circumference of waist after 2 years of Mediterranean diet. So we are now trying to see the effect of this with more detailed studies. And here I present the waist circumference change according to genotype, and also to the intervention group. As you may say, what happened here is that control group has the higher change in waist circumference. Before we see that all the people carrying this mutation, this alanine allele, they had higher waist circumference. But now we observe that there are differences between the waist circumference change in Mediterranean diet group and in control group. So what is your suggestion here? If you see this, Mediterranean diet is good or bad for these people? What do you think? Is good or bad? This is increasing waist circumference, no? So the effect is worse for the people in the control group. The people in the Mediterranean diet that has small change, no? Small increase in waist circumference. Are you understanding this? It's clear. So I just we can say that it's a protection to have a Mediterranean diet for these people that are carriers of this allele. They are protected because they have the allele, but they have lower waist circumferences because of eating Mediterranean diet. Okay? So we can see also this is the corresponding beta coefficient for this measurement, and we separate here Mediterranean diet plus olive oil and no mutation, or Mediterranean diet plus olive oil and mutations, and we see just the only significant component here is just control plus alanine carriers. And they have around 2.37 centimeter higher waist circumference when they are control group and they are not in the Mediterranean diet pattern. So this is the main finding with this mutation, that control people are in worse situation because they are not following the Mediterranean diet when they are carrying this mutation. So, now another gene variant. We study 4 genes. We have just commented the first one. But this is the second one. This is interleukin 6. As you may know, this is an important gene also that has to do with this association that now we know more and more about obesity and inflammation. There is an important relationship between obesity and a low-grade inflammation. And part of this important component is the interleukin gene. It's an interleukin gene. It's a protein, as you know. And what happened with this gene variant, that is in the promoter regions, sometimes is associated this C allele with a higher transcription activity and also higher circulating levels for this interleukin 6. And as you may know, obese people present high levels of this cytokine and is mainly also present in another comorbidities. So we want to know what happened with these people when they follow a Mediterranean diet. And we follow the same scheme that before. In this case, we study the outcome, the corporal adiposity, after 3 years of follow-up. And we have a total of 737 subjects, also in 3 groups. And we study mainly weight, basal, and after 3 years, we perform this analysis now with RT-PCR and allelic discrimination. That is a more sophisticated methods that allow you to genotype many people in less time. So it's convenient. And here I will present the basal characteristics of these people. Okay, we are now studying a polymorphism that is a chain of a guanine by a cytosine. But we study here the people that are homozygous for the mutation. Okay, they have 2 C allele. They are homozygous for the mutation. We have just around 100 people in this group and 600, more than 600 in the other group. And when we observe this group, this data, we just observe that there was higher waist circumferences in these subjects and also higher weight, little bit, higher BMI. So these people with the homozygous for the C allele appear to have more adiposity. Similar diabetes percentage, similar smoking habit. Okay. And when we study the change after 3 years in weight, we observe that these people in the Mediterranean diet, they have a higher decrease in weight, higher change than control on that Mediterranean diet and nuts. So we study also, but this is the mutation, and the CC allele people, they have higher, lower weight gain. Now here they have a loss of weight. They lost some weight because it's in the negative part, the change. And what happened here is that these people appear not to gain any weight, just to lose some weight. And when we combine just Mediterranean diet and this mutation, what we observe in these carriers people of the mutation, remember that before the people with the CC genotype, they had more adiposity. No? They had more adiposity at the basal state. But what happened after the interventions, after the 3 years of intervention, is that these people, they just lost some weight. They have lower weight. So this is a beneficial effect also of this C allele, because during the intervention the response was better in the people in the Mediterranean diet with olive oil and these C carriers people. So this is the beta coefficient, the regression model. And also we have the same observation that before, that the people with high BMI, basal BMI, has also the highest body weight change, a significant and opposite because of the negative weight gain. In this case, it's negative, so it's not weight gain, it's lower weight gain. And the people with the polymorphisms, as I told you before, when they had this polymorphism, they also decrease body weight. It has a decrease in body weight around 2 kilograms when they have the CC genotype. They are predict, because this is a regression analysis model, to have a lower weight gain. And when we study this with just the interaction term in the model, we study this now with the interaction. And we have age, all the variables that we adjustment variable, age, sex, basal BMI, diabetes, nutritional intervention, polymorphism, and also we have a new component here, that is the interaction between the polymorphisms and the nutritional intervention. And we see here that there was a significant, statistically significant interaction product term between the CC genotype and Mediterranean diet plus extra virgin olive oil. And what happened here is that the people, they are losing about 2 kilograms. So this gym diet interaction is associated with a lower weight gain. People are in a better situation when they have this mutation and they are also following the Mediterranean diet. And next gene is the adiponectin gene. As you may know, this is an adiponectin. Adiponectin is an adipokine that is secreted in adipose tissue that is present in our plasma. It's very abundant in plasma, has this globular shape, and there are many gene variants in this gene that are known in the promoter region, and also another 2 that we have mentioned before. And there are some studies, but we still have to confirm the role of these gene variants. Some of these are associated, the G allele, in this case, is associated with higher adiposity indices. Also the T allele is associated with higher adiposity indices. And maybe this is associated with protection against diabetes. But it's not very clear. As you know, human studies are very complex, and especially in genetics, we have many ethnical backgrounds, and this is sometimes very difficult to conclude for an effect. So in this gene, we study these 3 gene variants, and after 3 years of follow-up with the Mediterranean diet, we have also around 70 700 subjects in the 3 groups, as you know, and we study mainly body weight and other adiposity indices. And here we have the whole picture, okay? We have the 3 gene variants that we study, okay? And with the recessive model, AA versus C allele carriers. Okay? We have here also GG carriers and also TT, people homozygous for the mutation in the last 2 cases. Okay? And the number are here, and we study all the variables that we has just mentioned before, and we did not observe any significant differences in this table. When we study the body weight after 3 years of follow-up with Mediterranean diet in the groups, what we observe is for these SNPs, that is a change of a thymine by a guanine position plus 45. As you may see, we also have this significance, the effect of the basal BMI in the beta coefficient. And we also have here, that's the GG subjects with the 2 allele G, mutated ones, has a positive body weight, higher body weight. So there are some studies that confirm our results, and we go through, went through, and studied also the interaction. As before I explained to you, we have here in this table nutritional intervention, polymorphism. Now it's another polymorphism also in this gene, the adiponectin gene, and we have here the interaction term between nutritional intervention and polymorphism. Okay? And what we observe here is that, okay, as before we have seen the TT, here there is another polymorphism. In this polymorphism, the TT subjects, also the TT subjects have also higher body weight, around 2.5, 2.5 kilograms when they have this mutation. So this mutation is explaining part of the adiposity of these people. And regarding the interaction, we have a borderline signification for the interaction, the limit. And what we observe is that when these subjects follow a Mediterranean diet, the bad effect of the mutation is just lost. Okay? It's just lost. The mutation has a bad effect. But if you follow a Mediterranean diet, you are alive, you are better, because this is negative, minus 2.4 kilograms for people. The interaction, the beta coefficients explain to you part of this effect. So just finally we are now in the last gene that we study. This is the FTO gene. As you may know, it's fat mass and obesity associated gene. The protein that codifies this gene is demethylase that was discovered in 2002. It's present in many tissues, brain, adipose tissue, muscle. And it was discovered, this gene, through genome-wide association studies. That is a large study for genetics that allows you to study at the same time many mutations. And it was present, this mutation, in diabetes patients, but this gene was not explained the diabetics component of the subject. was explained the adiposity of these subjects. And also in general population, these are the first two studies with this gene. So it's a very recent gene, very recent studies, 2007. And all of them conclude that this gene is associated with adiposity, with BMI, with body weight, and so on. And we analyzed the effect of these polymorphisms that has this is the way we name the polymorphisms. Okay. And final 609 is very extensively studied because it's very important for humans. We also have 700 subjects. We performed the study 3 years, after 3 years of follow-up, and the magnitude to study was body weight, change in body weight. And what we observed were just initially, okay, we had 3 kinds of subjects: TT subjects, we had TA subjects, or homozygous for the mutation, that are AA subjects. And these A subjects, the alanine carriers subject, they have higher BMI, higher body weight. So adiposity has to do something with this gene. But what we observe is that after 3 years, the body weight change was negative for these subjects. They have the highest decrease in body weight. So it's a mutation that initially at basal state is connected to adiposity, but then you have better result after following Mediterranean diet. So we have here more detailed study. We separate the nutritional group, control group, Mediterranean diet. But what we observe here is that there were no differences between the Mediterranean diet people with regard to the controls. So the effect was more genetics effect because there was no differences in diet. And these people seems to be protected against weight gain, these alanine subjects. We include here TA subjects and also homozygous for the mutation, and for all of them the result was better than the people without the mutation. So having the mutation here benefits you against body weight gain. So here this is a summary, but maybe it's just to remember that this genetic variant has 2 effects. At least we study 2 effects. Firstly, we study the effects on adiposity, and later on we study the effects in regard to Mediterranean diet. Sometimes the mutation makes you to have higher waist circumference, as in this case. Other time, the mutation in this different gene makes you to have greater weight, BMI, or no change. And the important thing here maybe just to summarize the FTO gene, that this subject, the AA subject, has a more profound effect than when you are 80 subjects. So it's kind of dose-dependent effect for this FTO gene that is also reported in other studies. So this is a summary of the interactions between Mediterranean diet and genetic variants. And we have some interaction in PPR gamma just only for diabetics that I did not comment, just not to get you lost. And we found an interesting interaction between this mutation and Mediterranean diet. And we have this individual with greater weight gain after 3 years. And for the adiponectin, we have this contrary effect. After 3 years, lower weight gain in TT subjects. And for the FTO, we have no interaction between diet and this genetic variant. And just to conclude, we are in the trend, we are in the way to personalize nutritional solutions. We have to combine all these issues to learn how to perform the genetic analysis and to try to understand, and also in the future to have these individual recommendations. We have to understand the genetic makeup, all this polymorphism, to integrate the data, also with lifestyle, with nutrition, all the interactions. It's very interesting to address the interaction between genetics and nutrition. And eventually we will see these personal nutritions because we are different, there's no doubt, inside and in our genes and also in our phenotypes. But also we have to know that genetics loads the gun, but the environment that pulls the trigger. So we have to do a lot also. I just want to thank the PREDIMED people, Professor Martinez Gonzalez, who is the coordinator in Pamplona, in Navarra, for this study, also Professor Martinez, and Dr. Rathkin, who performed most of the studies that I presented to you today. And just to thank all the people that makes possible to do this study, all the dietitians and nurses and all the medical staff. And just thank you very much for your attention. And if you have any doubt or comment, I will be glad to answer.